Hello, General Biology, and welcome to the fifth in the screencast series for Chapter 9, Cellular Respiration. And we left off the last section talking about the Krebs cycle, and we had produced some electron carriers. We refer to it as being an energy extracting reaction. And we got electron carriers like NADH, FADH2. We also got some energy in the form of ATP, and we had some byproducts of carbon dioxide. And we're not going to worry about the carbon dioxide that was released, but we are going to focus on what happens with the NADH and the FADH2. So here's our electron transport chain. The electron transport chain is found in the cristae. So that's that internal membrane. And we know that, as I had said before, it's not the chloroplast, so it's not photosynthesis. We're not going to see any photosystems that will be present. But instead, we're going to have 3 ion pumps. So that's what you see here. These big 3 blue circles that you see are ion pumps. And we know, based on past experience, that ion pumps are powered by electrons. We've got some other transport proteins that are in here that are going to be just like stepping stones to help move the electrons from one ion pump to the other. And look at this, we have our ATP synthase, and we know from past experience that ATP synthase works by the process of facilitated diffusion. So that means that we have to get a high concentration of hydrogen ions in our space, that intermembrane space between the outer membrane and the cristae. Alright, so as I said, we're in that intermembrane space from the outer membrane of the mitochondria and the cristae. And this is the cristae membrane. So that this side that you're looking at is actually going to be the matrix where the Krebs cycle happened and the NADH and the FADH2 were produced. So the way that I'm going to explain this to you is first we're going to follow the NADH, the electrons from that all the way through, and then we're going to follow what happens to the FADH2 as a secondary electron transport. So here is our first ion pump. And NADH is going to drop off a pair of electrons to this ion pump, turn on this ion pump, and it's going to pull hydrogen ions against the gradient, going from a low concentration to the high concentration. We're not going to recharge our electrons this time around. The electrons are going to go into the next transport protein, go into the second ion pump, and it's going to pump hydrogen ions in against that gradient, going from a high concentration to a low concentration. Once they're done powering that pump, the electrons will move into the next transport protein and into the third ion pump, and they will power that pump to move hydrogen ions against the gradient and into that intermembrane space. So now, after it's been through the third transport or ion pump, it's lost energy, and the electrons have to go to the final electron acceptor, which is oxygen. And we breathe in oxygen, so oxygen is present. Also, if this was the plant, the process of photosynthesis provides the oxygen. So the electrons are going to join back to some hydrogen ions, which will attach to the oxygen and form water molecules, and water molecules will be released as a byproduct. That's why when you release oxygen from your mouth, you're also releasing water. If you've ever tried to clean your glasses and just breathed on them and got condensation, that's where that water comes from that process. All right, so that's the NADH. The FADH2 is going to drop its electrons off at this transport protein, and then it'll move into the second ion pump. So they don't start with the first, they start with the second, and that's going to move hydrogen ions against the gradient. Same thing that the first pair of electrons did. When they're done, they'll move into the second transport protein to the third ion pump, where they'll pump more hydrogen ions against the gradient. When they're done, they'll attach to hydrogen ions that will attach to oxygen that will form water, and water will be released as a byproduct. Now, in this intermembrane space, we have created a high concentration of hydrogen ions, which will move through the ATP synthase by the natural forces of facilitated diffusion. And as they move through this ATP synthase, it will activate the synthase to start to turn, and that spinning motion will pull ADP and ATP together so that ATP can be formed. The hydrogen ions will be pulled out, and they're going to be pulled back in by the pump. So we get some more ATP produced there. About 34 ATPs can be produced by this process. So that is the electron transport chain. So let's take a look at our totals. So from the processes of glycolysis, the Krebs cycle, and the electron transport chain, we get a net gain of 36 ATP just from that one glucose molecule. We really gained a total of 38 ATP, but we needed to start the reaction, so that's why we only claim 36. And as you heard from the Amoeba sisters, this isn't a set number, but it's under the perfect conditions that this is what one glucose molecule can produce as far as energy is concerned. Okay, we now have to look at the anaerobic process, and the anaerobic process is just a little bit different. So anaerobic respiration is in the absence of oxygen. So we're going to remove oxygen from our equation. We're still going to be working with glucose. Glucose will still break down and produce pyruvic acids, but depending on where this is happening, if it's happening inside the cell or if it's happening, we'll just say somewhere else for right now, will determine the product that's produced. But both reactions that we're going to talk about that you're going to see here and you're probably looking at and thinking, oh my God, Mrs. Saunders, I can't even begin to understand that. Both of them are going to wind up producing ATP, but a smaller amount of ATP, 4 ATP molecules to be exact. So it's not quite as efficient. So as I mentioned, there's two different types of anaerobic we don't call it respiration, we call it fermentation that takes place. And I want to focus on this one right here because your eyes are probably looking at it and saying, alcoholic? Wow! And you need to remember that alcoholic fermentation happens in baking. Every time we make breads, that is actually an alcoholic fermentation process that goes on. And it can be used in, and it's used in making wine and beer and different forms of alcohol, as well as it's the basic process in the start of making vinegar, some of your cheeses, your sour creams, nail polish, okay? Alcohol that we fuel our car with all happens with the basis of this reaction. So we just put it under a general category of alcoholic fermentation, but it's reactions that actually happen outside of the body in industry that we're looking at. So this other fermentation, lactate, you've probably heard of lactic acid before, And this is fermentation that happens within inside our muscle cells. And as I mentioned in a previous screencast, even though when you're exercising you're still breathing, the oxygen isn't diffusing in to your muscle cells rapidly enough. So your cells are going to have to work in an absence of oxygen for a little bit. And it still needs to be able to produce ATP in order for that ATP to be available for cellular activities. And you're still going to use glucose. Glucose is going to be the main endergonic molecule that will break down. So let's focus on that lactic acid fermentation here. Here's our glucose molecule. And just like we saw before, and this is just upside down to what we saw before, it's going to take 2 ATPs to break down this glucose molecule. And every time ATP breaks down, we get ADP and an independent phosphate group. Just like we saw in glycolysis, which didn't require oxygen either, when this glucose breaks down, we're going to get electrons and hydrogen ions that will be picked up by our electron carriers, NAD plus and NADH. So we will get that. That NADH is going to be used in order to convert the 2 pyruvic acids that were produced into lactate. And lactate, we know as lactic acid, is what helps our muscles move in the absence of oxygen and is what creates muscle fatigue, because it kind of fills in between the cells and creates gaps within those cells that push them up against the nerve endings, and that's why you feel like you have sore muscles in the process of exercising. So the NADH is going to be used as the energy source to break down this pyruvate into lactic acid. And we will get some ATP from this reaction. As I said, it only produces about 4 ATP. Alcoholic fermentation looks pretty much the same way. We start off with the ATP breaking down the glucose. We get the NADH just like we talked about before. It's used to break down the pyruvic acid, but depending on where the reaction takes place, the breaking down will turn into ethanol and carbon dioxide. So this is why bread rises. When you've got bread dough that you've made, you're getting ethanol that's being produced, and you're getting carbon dioxide, and that carbon dioxide is being trapped in that bread dough, and it causes it to rise, and that's why we need it down. So this is the process of alcoholic fermentation. And again, we're going to go over this in more detail in class, but this is just to give you an introduction to what's going on. When we see the chemical equations for lactic acid fermentation, this is what happens. Here's our glucose. The breaking down of this glucose can take 4 ADP, join them with 4 independent phosphate groups, and get this huge compound, lactic acid, and 4 ATP molecules. When we look at the alcoholic fermentation process, same thing, just a different place where it's happening. The above one was happening in the cells. This is happening in industry and baking. Here's that glucose molecule. Here's same 4 ADP, independent phosphate groups, but instead we get ethanol, carbon dioxide as a byproduct, and 4 ATP. I'm going to stop this here, and then we will conclude in the last screencast.